Hello, everyone. Welcome to this edition of the Modeling Perspective Speaker Series. I'm Eva. I'm part of the mathematical modeling team here at ECDC, and I'm very happy to introduce Tanja Stadler here today. She's a professor and vice chair at the Department of Biosystem Science and Engineering at ETH in Switzerland. She's a mathematician who's working on computational evolution of all kinds of applications, including epidemiology. And during the pandemic was part of the, and president of the National COVID-19 Science Task Force. And today she will be talking about the applications of genomics and modeling in epidemiology and public health. So I'll hand over to you. Have a nice talk, and after the talk, we'll have time for questions.
Great. Thank you so much, Eva, for the introduction, and thanks both Eva and Rob for organizing this. It's a pleasure to present today part of our work. And you should now see my slides. Okay. Eva, can you give me a just quick?
Yes, working perfectly.
Perfect. Okay. And so today what I want to talk to you is about statistics, modeling, and computational approaches we develop to decode pathogen genomes, understanding thus pathogen evolution and epidemiology, and also highlighting where that did and I anticipate will inform public health for evidence-based policy making. With this, to start, I mean, as we all appreciate, all life is characterized by the genetic code. So those 4 nucleotides A, G, C, and T. And this also includes viruses, which we could argue if they belong to life. However, each virus is characterized by such a genetic code. As an example, SARS-CoV-2 has 30,000 of those letters characterizing a single virion. And in our research group, we essentially leverage on this genetic code. So we sequence the organisms of interest. Today, we'll focus on viruses. And then based on the sequence of A, C, Gs, and Ts, we anticipate to uncover rules of past evolution of those pathogens as much as epidemiology, epidemic spread. And with, of course, the aim to also anticipate what happens in the future, which is relevant to public health. I'll start with highlighting how we do quantify pathogen evolution and epidemiology based on sequences which we obtain of the virus by looking at patient samples. So, for example, for SARS-CoV-2, if you went to get a swab to have a PCR test, from that swab, we may have extracted the RNA of the virus. So, of course, not any of your human kind of information, but the virus information, sequence that. And then today, in the first part, I'll explain what we can learn from that. I'll then have a few words on some thoughts on pathogen sequence data sharing, and then look forward how we can maintain genomic surveillance systems and potentially make them better and more robust. So first, I want to highlight or give you an intuition how genetic sequences can actually infer a transmission history. And classically, transmission chains, transmission history is very hard to reconstruct. If we have very good contact tracing data, we may, but typically we have no idea about who infected whom. And in the purpose of this talk, I'm not so interested if a person, Mr. X, infected Mrs. Y, but more how the overall tree looks like to infer parameters. So I'm interested on population level dynamics rather than individuals who infected whom. Here you see a toy transmission chain. So a horizontal line would be one infected individual. Say this is the first infected individual. I hope you see my mouse with the time going from left to right. And at some stage, this first individual may have transmitted the virus to a second individual, being the second horizontal line, etc. So overall, we see 4 infected individuals here. And assume this first individual was infected by a pathogen characterized by 8 nucleotides. Recall SARS-CoV-2 is 30,000, HIV 10,000. For simplicity that we can actually look at what's happening, we have here 8. And now the virus, as it replicates in the body of the different hosts, of course, there may be errors, copying errors, so-called mutations. And those mutations may fixate within one patient. And so, say the first patient at some stage, the C on the 4th position changed to a G. And in the second patient, from a T to a C on the 7th position, etc. We have no idea about those past processes, but what we can do is to sequence from patients who show up, say, at the testing center, and we take a swab. By now, from those 4 patients, we actually have the sequence. And hopefully you appreciate if 2 individuals are closer together in this transmission chain, there was less time to accumulate mutations. So we expect that their sequences are more similar compared to 2 patients which have a far distance on that chain. Say, for example, this top one is very loosely related to the very bottom one, and they have 3 mutations on their path. So they differ by 3 positions, while the 2 very close, they only differ by 1 position. So based on sequencing and then essential clustering, more similar sequences closer together in a tree compared to the more different sequences, we can reconstruct from sequences essentially the transmission history. And that inference is also called phylogenetic inference. We infer a phylogenetic tree, we do phylogenetics on the sequences, reconstructing the past tree relating the sequences. As I mentioned, I'm mainly interested on a population level, so I want to know, what does this tree now tell me overall about the epidemic spread in the population, say the epidemic spread of SARS-CoV-2. So we want to learn from this tree what is, for example, the reproductive number, so the number of secondary infections caused by a single infected individual, which is a key quantity in epidemiology. And essentially, for a second, suppose this transmission tree is complete, so we sequence everybody who was infected, and we can reconstruct who infected whom perfectly. We can just count the first individual infected 2 people. If not, this tree will be bigger, and we always see one person, one horizontal line has 2 offsprings. Well, then the expected number of offsprings is 2. So the reproductive number would be 2. There's a note of caution. So first, we don't know who infected whom. So actually, we don't know that this bottom individual infected a top individual. It could also be vice versa. Plus, we have typically a very sparse number of individuals sequenced. So by far, we don't have all the individuals included in the tree. So what we're doing is we develop statistical methods to first integrate over who infected whom. So is it the bottom one infecting the top one or vice versa? And integrate over potentially 99% of the individuals we have missing. So we only include 1% of infected individuals. Doing those statistics and essentially this integration over missing events and missing knowledge, we can quantify the reproductive number essentially based on the sequences by, as an intermediate, reconstructing the phylogenetic tree. And in what follows, I want to give you some examples how we did that for different pathogens in the past. If you're interested in the exact math, how to do those calculations, how to integrate over unsampled individuals, unknown direction of transmission, we actually just in May finished writing a book. It's called Decoding Genomes. It's a whole group of people, past and current members of my research group. And in that book, you can find detailed explanations how this inference works. It is really going from sequences to phylodynamics. Down here, you see the link, decodinggenomes.org. The PDF is freely available, so you can just download it, have a look. Hopefully, you find it useful and interesting. There's also a feedback option in case you spot something you want to report back or have suggestions. You're very encouraged to use that feedback option. If you would want the book to have it in a printed format, also on that link, you can order it. With that, let's go to data examples. So in 2016, we decided we that we want to understand better seasonal influenza, and in particular how it travels around the world and into an average city. And as average city, we chose Basel, where our department is based. It's a smaller sized city, 200,000 inhabitants, but the bigger area, roughly 500,000 people. It has a small international airport and international companies. You may know Roche, Novartis, so big pharma. And so we want to understand, is influenza, seasonal influenza, driven in a city based on large local transmission chains, or rather based on introductions? And in red, you see the sequences we obtained from patients in Basel who showed up with influenza-like illness, who agreed to participate in our study, and actually who we then tested and confirmed to have influenza. We sequenced those samples. And those samples, the red dots, fall within this bigger tree, and that's really a global tree of influenza, so samples taken from around the globe. So I hope from that tree, without showing further stats, you appreciate if we sequence in Basel influenza, essentially we obtain the diversity of the world, meaning there are tons and tons of introductions from anywhere in the world into Basel, which is not, as said, this biggest international hub, but it has international connections. So meaning influenza travels around the world very fast and mixes a lot, so that in our city, we see that pretty much we have strains from anywhere. And this is now a side comment, this study already made us a bit skeptical that travel bans during the pandemic help too much, because essentially once we look at it, the virus is already everywhere. This is all phylogenetics. As I was saying, we're in particular interested what we can learn beyond looking at the tree, the dynamics. So we estimated the reproductive number of seasonal influenza within Basel. You see here at the beginning of the outbreak, it's well, above 1, no surprise, otherwise we wouldn't see an outbreak. Maybe around 1.2, 1.3, but rather large uncertainty intervals. Those large uncertainty intervals were bigger than we anticipated based on sample sizes, and it's due to we have so many introductions and thus actually very small transmission chains. And so we don't get too much signal on what the precise reproductive number is. We were then discussing if we should increase our sample sizes to get actually the dynamics a bit more confined, to also investigate if the reproductive number rises or falls with school holidays, temperature, humidity. But before we could actually embark doing a much bigger influenza study, we obtained a lot of data for a different pathogen, and that was then SARS-CoV-2. So early 2020, the world appreciated that the pathogen is spreading globally. And based on the first sequences, first, of course, from China, but then also from Italy, Washington State, and then this cruise ship, the Diamond Princess, where very early on there was an outbreak, we inferred phylogenetic trees for those 4 different regions. And from the trees, then the reproductive number, here called R0, because it's the reproductive number at the start of the outbreak when everybody was susceptible. What we inferred was for the 3 geographic locations, the reproductive number was somewhere between 2.5 and 3. For the cruise ship, slightly higher. That's not a surprise. People have different living conditions there. They are much closer together, so we anticipate it to be higher. And I want to highlight here in a statistical method, in particular, we use a Bayesian approach. In a Bayesian approaches, if you're familiar with them, you have a prior, and our prior was lower. So there was signal that the reproductive number is well above 2. In parallel, colleagues of us in Bern used confirmed case data to estimate the reproductive number and also got to something 2, potentially higher, albeit also with a rather big variance. Taken together, our study and other studies, the reproductive number was estimated between 2 and 3. And now when you recall that influenza was maybe 1.2, one can appreciate that the spread early 2020 was quite fast. And in particular, it means based on very simple epidemiological SIR models that with such a fast spread, only when more than half the people will have been infected and recovered and are immune for reinfection, the pandemic will again decline. And of course, in a decline, still many people get infected. So essentially, the vast majority of people will get infected unless there's measures to be taken before a vaccine becomes available. And I should highlight all of that was done before any of our countries went into lockdown. In fact, we published on a blog post this graph the day when the first case of SARS-CoV-2 in Switzerland was confirmed. So when life was pretty normal still, and then only the weeks to follow did the big measures, were the big measures implemented. Anticipating, among others, based on those results and what people and colleagues globally observed, we were essentially anticipating there'll be many, many cases around the globe, but of course also in Switzerland. We're well connected, as we saw with the influenza. So we set up a SARS-CoV-2 sequencing consortium in Switzerland. It was a large number of people was involved. We started sequencing in late spring 2020 on a weekly basis, SARS-CoV-2 genomes. And here you see the phylogenetic tree of samples from the first year of the pandemic. This was really a research project. At the time, Switzerland didn't have a national sequencing program, so we did that as part of a research project. And in fact, Sarah Nadeau, who was a PhD student at the time, she then used on our sequencing data, again, phylodynamic methods, to estimate the reproductive number in Switzerland based on sequences. And without going now in detail what the exact dynamics were, I hope you appreciate if we compare that to confirmed case data, the patterns are quite similar, though confirmed case data, because we had so many confirmed cases then, had lower uncertainty intervals. You might now think, why even bother with the sequences? Just use the confirmed cases and we get more tight uncertainty. If it's just about the reproductive number, sure, but I'll get in a second to an analysis where we need the sequences before those saying that actually those R-value estimations we did, and we did those both for Switzerland, but also for around the globe for confirmed case data. And in Switzerland, at some stage, they even got into a law, were used by the politicians. So actually restaurants were closed or open based on the R-value being above or below 1. So why we estimated a reproductive number based on sequencing data was twofold. First, compared to confirmed case data, we can actually correct for introductions. So we see in the big phylogenetic tree using Swiss data and data from abroad what the Swiss transmission clusters are, so we don't confuse introductions with within Switzerland transmission. And so we can be quite comfortable to trust those R-value estimates. And I should say here, green and orange differs by making a different assumption how we define what a within Switzerland transmission chain is, and it's essentially a robustness test. If both agree roughly, we are quite confident that we are not doing something too crazy when we make assumptions what part of the huge tree is really a Swiss transmission chain. We wanted to investigate in 2020 if the contact tracing, which was slowly but surely implemented in Switzerland, if that actually had an effect. And what we were assuming is there was a reproductive number, but for each Swiss transmission chain, that might be a little small chain with 4 individuals, say this chain and another chain, and maybe some chains with 10 individuals or 20. We assume once we know about the first case, so the first sequence, the transmission rate, and thus the reproductive number, may have dropped because contact tracing could have kicked in. Importantly, Swiss transmission chains, they start from an introduction from abroad. And at first, we don't know about this introduction. We only can know about a transmission cluster once we identify the first infected person. So where the purple box starts. And so now we assume there's some parameter, say S, a dampening factor, and by maybe 50%, the transmission rate is lowered upon identifying the first case. And that, if that is the case, that would indicate that actual contact tracing may have lowered the transmission rate by 50%. I want to highlight here, we're not doing a causal analysis that we're sure it's the contact tracing. All we can say, upon seeing the first case, we have a drop by 50%. However, we didn't see too many reasons or hypotheses what other factors could have caused a drop upon seeing the first sequence. So any drop we see in the analysis, we explain with contact tracing efforts. And what were the results? Essentially in spring, in this first part, data was too sparse to have any certainty in results. So we don't really know. In summer, depending on the assumption what a Swiss transmission chain is, we see roughly 30 to 60% of dampening. So we see a severe drop in the reproductive number. While at fall, then we essentially see 0 to maybe up to 15-20% of dampening of the reproductive number. And this big dampening in summer also coincides with contact tracing having been installed in Switzerland, and we had low case numbers when contact tracing actually could be effective. While when we saw no effect in fall 2020, we had a huge second wave. And in fact, the contact tracing centers said they're completely overwhelmed. They can't even process all the cases they get reported. So in that way, we interpret our study that roughly between 35 and 63% the transmission rate and reproductive number was dropped through performing effective contact tracing. So then post-2020, actually the Swiss Federation asked for having a broad sequencing program in Switzerland to track SARS-CoV-2. And with the increasing numbers of sequences becoming available, the data handling also became more challenging. So now we have 18 million SARS-CoV-2 sequences globally available. You cannot use just standard databases and encryption to have and expect that you have fast access to the data. And that's where Chao and Chen came in. He just started back then a PhD in the group coming from computer science databases. And he implemented a way to actually host, and currently we have 18 million SARS-CoV-2 sequences globally, compress them effectively, so store them as you would store the size of the data as with classic compression schemes. However, he uses a compression or developed a compression scheme where essentially per column of the alignment, you condense your data in a way that all information remains and you can still query your data. So you can still the compressed data query and get in milliseconds answers about, how many sequences in Switzerland have mutation, X on site 4 and mutation Y on site 10. And he implemented all of that in CoVSpectrum, which I invite you to explore, covspectrum.org, and you can explore different pathogens. And actually, the early version of that we used when alpha started spreading at the end of 2020, just before Christmas, the UK reported that there's a new variant. And in Switzerland, we were just out of this terrible second wave. Case numbers were, in fact, on the decline, but we saw this proportion of alpha in our sequences going up. So roughly 2% of the sequences by Christmas were alpha. Based on mathematical modeling, essentially you have two competing variants, alpha and the wild type. So, and you can say alpha has potentially some fit disadvantage. That means they compete. How alpha should become dominant would be a logistic growth model. And we estimated that alpha has a transmission advantage of 40 to 50%, and thus will become dominant in March and would cause another wave. The size of the wave, of course, depends on how many contacts we have and thus how much transmission is possible. And in fact, based on such considerations, our government decided when case numbers were really dropping nicely in mid-January that they strengthened the measures. This was based on realizing or appreciating that later alpha will be dominant, and that's what happened. So this is empirical data. It did become dominant. And so at that time, the government really took the underlying modeling serious that there will be a problem in March, which was, of course, then also challenged to communicate to the population. Why? If numbers go down, you have stricter measures than when you had increasing case numbers in the second wave. So those were some examples of analysis we did on the genomic sequencing data during the pandemic. We realized throughout the pandemic also that the global data sharing is not working as well as we would hope. And here I should start off with technologically. As I said, we never had millions of sequences for a pathogen prior to SARS-CoV-2, and new database solutions were needed. And so Taran implemented the database solution behind COSpectrum. And we, meanwhile, in a bigger effort across Switzerland, together with the group of Richard Neer, Emma Hartcroft, but also Theo Sanderson from the UK, and Artem Babayan from Toronto, and all their team members, we put approaches from our database, which we use in COSpectrum, and the API, which we call Lapis, like a lightweight API. We combine that with Nextclade pipelines, which are within the NextStrand project, which you may know, essentially processing sequencing data before putting them in a database. We combine those efforts, make nice frameworks around, apply the compression schemes I was talking about, which we have in COSpectrum. So, and have now open source software to share genomic sequencing data. So you as the user can set up your own genomic sequencing database, and people can then, deposit data there, can access data, you can fast forward your data. This can be used for having an open database and share, with the world, or if you have sensitive data, say for HIV, colleagues are now interested to use the software to host internally some HIV data where for privacy reasons they can't share. And so this is an open source software. I invite you to check it out, loculus.org. Essentially for any pathogen, you can set up your instance and share data through that instance and easily query all the data you host in a database. This is used in a first bigger attempt, this software solution by Pathoplexus. And Pathoplexus is a new open source database dedicated to efficient sharing of human viral pathogen genomic data with the aim to foster global collaboration on public health response. We started with 4 viruses for which we believe there's not a proper home where people share that. That's the Korean Congo hemorrhagic fever virus, Ebola Sudan, Ebola Zaire, and West Nile virus. This database is run by an international executive board from the community for the community. You can look on pathoplexus.org who is involved. Just very briefly, our executive board, it's 5 people, essentially from 5 continents, each continent 1 researcher. And in that way, Pathoplexus, the vision will be that it serves the community, both data submitters and data analyzers. Second, a core aspect in the governance system is that everyone has access to the data. And in particular, you can also have programmable access. So if you have a dashboard, you can automatically feed in the data and show them on a dashboard. So now you can ask, is that different from like making your data completely open right away? Yes. Well, for one, we offer very easy data submission, but you can ask for 1-year restricted period where third parties cannot publish as a scientific paper your data, but everyone can use it for public health. So doing some analysis, share them on social media, share them in reports to your government. That's allowed. Just one is not allowed to write a scientific paper for a year, so that the researchers who initially produce the data can write their own paper. After a year, the data automatically becomes open. So the service is that you don't have to fiddle around with ENA. We automatically, or Pathoplaxis pushes it to ENA. And then for data generators, important, sequences get a DOI to be citable, as we envision, or we strongly feel that data submitters should have more quantifiable references to their efforts to the sequences, so that sequence data is also a citation and not only the paper. Please play around, pathoplaxis.org. Again, there to the Pathoplaxis organization. If you have any comments, feedback, feel free to contact them. So I want to end with genomic surveillance and epidemiology going forward. I think even though globally we cut down a lot on sequencing, we always need some virus genomes from patients, not just SARS-CoV-2, but more generally. I'll quickly highlight an effort we are pursuing there. And then another level, on a population level, I believe we can leverage more on environmental samples where we get averages on populations, for example, through wastewater. And I also want to show you one example. First, on patients, what we started now doing, instead of analyzing the patient swabs for SARS-CoV-2, we take swabs from patients with respiratory infections and use a so-called hybrid capture sequencing panel where there's 30 different pathogens. And while, for example, we did a pilot before from Switzerland, there was just one RSV sequence available. In this pilot, we already have 70-some sequences. And similarly for other understudied viruses. We now, and also colleagues in the UK, want to go ahead with such panel sequencing of respiratory patients with respiratory symptoms to better understand the less studied viruses and anticipate what their dynamics is and learn about their epidemiology. From the community, rather than sequencing tens of thousands of patients, we can also look in wastewater. And what we're actually doing is every hour, automatically a bit of wastewater is collected. And that is around 14 different geographic locations in Switzerland. The samples are prepared. The concentration of virus is quantified with PCR. We sequence, and then we can characterize variants, anticipate waves, do modeling. We currently do that for SARS-CoV-2, influenza, and RSV, and mpox and seasonal coronavirus is going on as pilots. Then, and Adrien Liaison, a PhD student group, was spearheading that. We can estimate reproductive numbers based on this measured data, essentially as we did on confirmed case data for SARS-CoV-2. And not only are we doing that as shown here on SARS-CoV-2, but we do that across those viruses measured. And so in the last winter season, it was actually an excellent way for us to see and anticipate where we have waves of a particular pathogen building up based on looking at a few wastewater samples rather than thousands of patients. With this, I just want to summarize that I hope I conveyed that based on the SARS-CoV-2 experiences, leveraging on sequencing opportunities can lead to real-time insights into pandemic spread. And in particular, based on those couple of instances I highlighted in Switzerland, I guess after we had some trouble initially talking between scientists and policymakers, later the exchange was actually going quite well. Meaning I think there's opportunities to now expand our surveillance networks beyond SARS-CoV-2 and maintaining a well-functioning policy interactions. This, though, comes, of course, with challenges. First, and most countries cut back enormously on monitoring pathogens. Do we find enough money for that? Do we maintain and establish for understudied pathogens timely data sharing? and how do we maintain the dialogue with the policymakers now that other crises are more, more often in the news than pathogen threats. I want to thank the whole group who did the fantastic research and all our funders. And thank you. And I'm very happy to take any questions you may have.
Thank you so much. We already have a first question. Till Krüger, feel free to unmute and ask your question.
Yes, thanks, Tanja, very much for the nice talk. I have a question to the beginning when you showed this outcomes of the R0 estimations. Did you ever try to separate out the in-household and the out-household infections? So I'm asking because in R0 makes not so much sense for inside household because you have saturation effects immediately, right? And we are in a confined area. And so it's very interesting to look what is really in out-household R0. And then it's in a non-trivial way combining with the household structure. And yes, the better measure is the secondary attack rate, which is frequently measured in empirical studies. But you need a bit sophisticated models actually to do this, right? You cannot do it with a compartment model where you don't have households in. So the question is, but you have seen you have done it for contact tracing. So you have maybe also used more complex models than simple ones for the R0 estimations here. So my question is whether ever you looked in this direction.
Yeah. So we discussed that direction a lot, but we did not use a complex model within household, between household. However, what we did was we subsampled our sequencing data to hopefully ensure we don't have from one household more than one sequence. And the reason we didn't go for more sophisticated models, well, first, it's not trivial for phylogenetics, but second, and more importantly, mostly we had sequences, we just knew the date and the location of sampling, but not kind of, are they the same household? What we then did is, if on the very same date we had in a, close by the exact same sequence, potentially sampled at the same hospital or the same kind of practitioner, we just assumed, okay, those identical sequences are potentially from the same household. So it's a very crude way to, and so we subsampled pretty much all available sequences, having in mind or having the goal, which I'm not saying we achieved always with those crude measures, but having just one sequence per household.
I think this is a very interesting because we have done with a more complex agent-based model here for Poland in the initial phase this. And if I correctly remember our estimations of the out-household R0, if the sequencing data you showed is essentially without the household, pretty much are in the range of the values you showed here. So that would be interesting to have a closer look. Yeah, and definitely with sequencing, you can maybe somehow guess what's the household was indeed. Thanks a lot.
Are there any other questions? There's something in the chat. Compliment for the book. The book is really incredible. Someone says, if no one else has a question, I maybe have a question. There is a lot of sequencing data out there that is, you've showed that there's a lot of efforts in trying to get this more accessible. What do you think is the sequencing data for epidemiology is underused, perhaps? And on the other hand, what is the data that is still truly missing for answering the questions that are relevant?
Right. So I would say underused for SARS-CoV-2, potentially, in retrospect, we're always knowing more, but potentially I would say when Delta was circulating, yes, we invested a lot of money in sequencing a lot of cases of Delta. And how much did we analyze them? Well, we did analyze them. I mean now the global community, to then in a timely way see how fast Omicron takes over from Delta. That could have probably done with fewer sequences, and many of them are potentially underused. And I'm also not so sure, to what extent we want to look into them. But this is a very particular, we were in a pandemic and people also invested a lot in sequencing. I would say out of this pandemic now, what we are missing is reliable, continuous sequencing. So say with influenza, I had a research project. So for one season in Switzerland, now you have, quite a few influenza sequences. And then maybe 2 years later, a colleague did something similar. But this very continuous sequencing. And then influenza, you could say, because of the vaccines, that's from the WHO still supported. So it looks way better than RSV. As an example, in Switzerland, we have one sequence. And now with the vaccines becoming available, the monoclonal antibodies becoming available, we actually should look at those sequences. And I'm not saying like in a pandemic sequencing now millions of samples, but just a number, which of course for each geographic location we would need to figure out how many, but study all those different array of viruses. And that also would prepare us if we see very weird, clinical phenomena to actually link back. Could that be due to some pathogen wave, say an adenovirus wave, which currently, we may miss?
Very, very good points. Something to keep in mind also here, I guess. Does anyone else have any questions, comments? Also feel free to write in the chat, unmute yourself, just ask something.
And maybe just to also add there, because you asked about how much of the sequencing, I think for just an average, like a population average, I do think environmental samples and wastewater is a very convenient one, can bring us a long way. And at least for us here, it's now so much more cost effective than sequencing thousands of patients. But it depends then what the question is. Of course, the lab people, if they want to explore properties of the virus, they need the virus from a patient and can't deal with the wastewater. So it needs both, but we could find a good strategy with the least costs.
There's nothing else. Then we're very nicely on time with 45 minutes. Thank you all so much for coming. Thank you, Tanja, for the amazing talk. And I guess for the next version of this series, you'll receive an invite if you're on the right mailing lists.